I'm Tom. So I am a researcher at the University of Sheffield in Salisbury, and I'm an experimentalist really. Primarily look at DNA and DNA protein interactions with AFM. But a big part of what we do as well in our group is image analysis and trying to quantify what we're seeing at the edge. So this kind of morning is going to be an introduction to image processing of SPM and AFM images. So the first sort of maybe hour or so, we'll do a bit of a talk about the various considerations that you need to have when you're processing SPM images. And then the second 2 hours is going to be a little bit more hands-on with Gwydion, which I'm sure some of you will have heard of already, which is an AFM analysis software. And then a little bit of an introduction to automated image processing, which will be very useful for all of you, I'm sure. So let's get started. So kind of the very broad question of what actually is image analysis? To consider a pixel is actually derived from picture elements. So each pixel in our image is just a number. So we kind of go into this drawing here. We see that each pixel has been assigned, in this case, a value from completely white to completely black. So every single pixel within our image contains data, contains information. And in image analysis, we want to be able to extract that information. And why is it important? So as I said, because each pixel contains information, we can use that to extract statistics from our images. So this is an image of some cells, and each of these cells has characteristics that we might want to be able to measure and want to be able to quantify. So we can measure the sort of area and the perimeter and things like that of these cells, and we can look at differences between different samples. So, for example, cancerous cells versus healthy cells, and do these measurements that we make change between the two samples, and can we quantify that to learn more about what we're actually looking at? So there are lots of tools in the community, in the bioimage analysis community, for image analysis. I don't know if many of you have heard of this, but there's an image analysis community called Image.sc, and they have a whole kind of range of tools within this community. So you can see, I think, ImageJ is on there, Fiji is on there, Cell Profile is on there. It's very optical image based right now, but they can do lots of things such as denoising images, segmentation of certain areas of your image. So if you went to the Brooker workshop yesterday, you'll see something like this that the AFM's doing now. And from all of those things, we can extract different features and visualize that data. So there's a load of really good packages out there in the community already for image analysis. But what about AFM image analysis? So it is quite different to standard microscopy. Generally, with a standard microscopy image, you would get an image format file, so you get PNG, a TIFF, something like that. But the AFM data is very different in how it looks. So it's not just an intensity image, it's not just pixel values of intensity, it's actually topographic height map. So just in the case of a normal topographic AFM image, you also get all of this 3D information. And then you add on top of that, when you're taking full scale data, you've also got all of that information as well. So it's really, really rich data. There are also quite a lot of different artifacts which I'll come on to need to be considered as well. It also spans quite a broad range of applications. So these can be used from sort of in physics and material science, looking at things like semiconductors, looking at battery technologies, looking at cells, DNA, So there's a whole range of different applications to AFM, and that also brings it sort of different challenges when thinking about how to analyze images. It's also quite limited in its data availability generally. With, again, optical images, you can take thousands of images, and when you're thinking about deep learning models and things like that, it's quite easy to train data on those thousands of images. Whereas AFM is generally fairly low throughput. It's quite a slow technique, talking 5 to 10 minutes per scan. It's quite difficult to gather enough data for your statistical analysis or your sort of AI analysis. So that's, again, something that needs to be thought about. And the big thing, and the AFM community in general is getting better at this, but historically it's been very bad for proprietary file formats from various companies, making analysis between different software is very difficult. It is getting better now. People are generally adopting the SPM image format, but yeah, it used to be quite bad for that. So it isn't a simple process to analyze AFM images. This is what happens when you drag an AFM image into ImageJ. So using a sort of very standard image, very widely adopted image processing software, just that a lot of nonsense because of how differently formatted the AFM data is compared to a standard image. It also comes with a lot of different considerations. So these are a sort of range of image artifacts that you really have to take into account and really think about when you're processing and analysing your AFM images. And I'll sort of go into each of these in a little bit more detail, but they're very unique to AFM. So these are a lot of image artifacts that you wouldn't really come across with other techniques. So just to kind of go through the very basics of an image processing pipeline, really, with an AFM image. So this is the kind of raw AFM image. This is absolutely straight out of the AFM without any post-processing at all. This is what your image will look like. This is a topography, this is a height image. We can see here that there's kind of a big tilt across the surface. And that's very simply just because the plane of the scanner and the plane of the sample aren't perfectly parallel. So one will be at a slight tilt to the other, and that just gives you this large tilt over the surface because nothing is ever completely flat. So that first thing that we need to do is actually remove this, so we can flatten this out and remove this tilt. And this is usually just done with a first order polynomial plane subtraction. So you just fit a polynomial to that first order polynomial and then just subtract it from the image, and that flattens this large tilt that we see. So you might notice that there's still quite a kind of unevenness across from sort of top to bottom of the scan. And this is actually due to variations in the feedback loop as each line is scanned. So the feedback loop in the AFM isn't perfect. So as it scans each line, it's a raster scan, as Alex was talking about yesterday. As it moves down to the next line, it doesn't always get the height position exactly in the same place as it scanned the first line, which means that each line is then given a slightly different baseline height value, and which is different from the rest. So you can kind of get this sort of stripy effect down the image. So again, that's something that we need to correct for. And kind of remove these height variations. And again, this is done with kind of fitting these lines and finding the mean value in some way. I'll go through sort of specifics of that in a bit, and subtracting that from each line. So then each line is equalized with the rest of the lines to get a more flat looking image now at this point. We also have an artifact in AFM images called scars, and it's probably very difficult to see on the screen. But scars come from the AFM tip dragging or skipping across the surface. So when it encounters something quite high, so as your tip's moving along, it encounters something quite high, it can sometimes hit that and skip a few pixels. It'll just miss the next few pixels that it's touching the surface. And it usually comes up on the scan as either a completely bright white line or a completely dark black line, which is missed pixels. There are some ones that you can't really see it on the screen. And again, we need to be able to remove those because they sometimes look like data. So you've kind of got a bright white line, they can be quite thick as well. It can sometimes in our case look like the DNA looks. So it can be mistaken for the data that's actually that we actually want to look at. So those are some things that we need to remove, and again, we can do that. And another big thing with AFM data is that it's quite noisy generally. It's a mechanical technique, it's not optical, and it's very heavily influenced by thermal and electrical noises. And that can introduce a lot of high frequency noise within your data. And you can apply various filters to then remove this noise and smooth the image out. And that makes the features a lot more, kind of a lot clearer within the image when you remove that high frequency noise. And then once you've got your nicely flattened, sort of pre-processed image, you want to actually be able to quantify what you're seeing. So you want to be able to extract features. So we do this by masking. So we try and identify the molecules of interest. This is just a height-based threshold to, in this case, identify the small DNA molecules that we see in our image and separate them from the background. We can then apply various steps. So in this case, we're really only interested in these DNA molecules here. We're not interested in the tiny little bits of contamination in the background. So we can then take our mask and apply certain constraints on it. So we can look for anything that is above a certain size, below a certain size, and remove all of the features that we don't particularly care about. So then we've got just our data that we're interested in. And yeah, we can start to try and quantify what we're seeing there. And I'll kind of go through a bit later, kind of the things that we can pull out of these images. So I'm going on to image artifacts. Like I said, there are a lot of different image artifacts with AFM that needs to be thought about. Biggest one is drift. So this is basically a sort of slow movement of various parts of the AFM. So whether that be the laser sort of slightly drifting across the detector, the position of the piezo slightly drifting as you're scanning, and you basically can just elongate features. So you're basically dragging slowly across the surface. So this is what your image should look like, which is a series of dots, a series of blobs. And if you get a lot of drift, it drags everything out and makes everything look like rods. Now, this is something you can only really compensate with by allowing your system to equilibrate, everything to sort of settle before you start imaging so that things aren't moving around as much. The important thing to be aware of here probably is that you need to think about what your sample should look like. Because if you just got this and that's all you're seeing, you would think that that is what your sample looks like. And you've got to be really careful. You don't get tricked by an image artifact into thinking that's actually what your sample looks like. And then go and publish that, and that's what you think it looks like. And in actuality, it is more like that. So yeah, this is kind of one of the big things that that comes up quite regularly is drift. You also have, this is quite similar to drift, but you have creep, and this is more to do with the scanner. So how the scanner moves, it can be slightly non-linear as well, and you get these edge effects where the edge of the image is elongated or compressed because the scanner isn't moving completely uniformly across its range of motion. And this one is easier to correct for because we can do calibrations. So this is the calibration grid. Each of these little squares should be about the same size. And we can image them quite regularly on our systems. And if they do look like what we see on the left, we know that something's going wrong and we can recalibrate the scanner to make sure that it's not. So ringing is another effect we see. So see this around. So these are some cells, but you can see these kind of weird blobs around the cell. And this is ringing. So this is where the feedback loop that we use, the gain on that is set too high. So you're basically amplifying your signal, but you're also amplifying a lot of the noise, and you get a really high noise background basically. So you get a lot of, this is all just noise that's being amplified by the gain being set too high. And again, this is something that needs to be considered during imaging, but also we need to be aware of when we're processing our images, that this doesn't kind of influence what we're seeing. Also have effects such as parachuting. So if we set the gain too low, find that, it demonstrates this quite nicely here, as the tip is moving over an object, it doesn't quickly come back into contact with the surface. So it can go over an object and then it starts very slowly responding. And it gives these sort of effective elongated features within our sample. So this is what I think Alex referred to yesterday as tracking. So if we track well, we move up and we immediately move back down again, and everything looks like a nice circle, like just here. If we're not tracking well, our tip doesn't remain in contact with the surface as we move over features. And this can be, again, adjusted by increasing your gain, increasing the force that you're pressing on the sample with a little bit as well, to allow it to sort of remain in contact better. And this probably is the biggest bane of most AFM, especially when you're doing high resolution single molecule AFM, is a double tip. So it occurs really when either the very end of the cantilever, so that very sharp end of the cantilever, can split slightly, and you get essentially two cantilevers like this, or you get something attached to the side of your cantilever, so you have a blob that's on the side of your sharp cantilever. And when that happens, you essentially see a double image of everything, because each of these tips interacts with your sample. So you can, kind of both are getting a false response, both then register within the image. And actually in this image here, we have 3, maybe 4 repeats of each molecule. So there's almost 3 or 4 tips within your tip. This one is almost impossible to correct for. It would be very nice if we could correct for it in some way, or at least recognize when it happens. So that's maybe something to think about in terms of thinking about image analysis methods, is whether we can actually detect presence of a double tip. And yeah, these are just some other image artifacts that are quite common. So what we're seeing here, I mean, when have you got a very repeated sort of artifact occurring in your image? So for example, anything looks like a triangle here. We don't think our samples should look triangular in any way. It's usually an indication that the end of your tip has somehow, rather than being a point at the end, it's now a triangle at the end. And it's making everything look like a triangle. And in this one here, you can see that each of these molecules should be looking like this. Have got a little thing, a little kind of triangle again associated with it. And that again means that you've got something stuck to the side of your tip that is creating this artifact next to your molecule of interest. This is also quite a big one, and this, again, I think Alex mentioned this yesterday. But tip broadening is kind of a big problem, especially, I think it's more so a problem when you're looking at small single molecules such as DNA. Less of a problem if you're looking at materials. But as the tip gets blunt, basically broadens out the features that we see. So rather than a really nice, sharp kind of top hat there, it slightly broadens out. If your tip gets even blunter, that broadens out even more. It's not too much a problem for high features, depending on the thickness that you actually want to see. But for trenches, it can be a real problem. In this case, you can see that you can't really get down to the depth of the trench because of how fat your tip is. And we always need to consider that, the width of the object that you're seeing is always a convolution between the actual width of the sample and the width of your tip. So kind of see that here. This is the width of your actual object. This is the width of your tip. And it's a kind of a multiplication of those two that gives us our actual image width. And this is true for sort of circles and pillars. And actually, there are equations that you can work out from. You can kind of back out whichever way you want to go. You can either go from the known width of your sample and the measured width of your sample. So for DNA, we know that DNA is 2 nanometers in liquid, is 2 nanometers wide. And if we measure it as 6 nanometers wide, for example, we can then use this equation to actually work out our tip radius. This is starting to think about tip deconvolution, which is a big, really big challenge in AFM, is actually knowing how wide the tip is and knowing from that how that's affecting your imaging. And yeah, it's historically, it is quite difficult, especially when your sample isn't a very defined width. So if you're imaging something like a kind of meshy network that all of the strands have different widths, is quite difficult to actually sort of define it. Whereas, yeah, certain samples you can define a lot better. So yeah, as it says, overall, with image analysis, it isn't simple for AFM. But if we take dedicated data handling and image processing software and considerations, it is possible to actually convert that into decent data that we can quantify. And the last sort of thing I want to say on this before we actually start doing some image processing ourselves, I think that's probably slightly more interesting than hearing me talk about it, is that bioimage analysis on the whole is a very collaborative activity. So we can't just do it in isolation. We need to work well together between experimentalists and computational people. This is always an argument we're having in our lab between the two sides. Yeah, the coders always want us to take better data. We always want them to write better code to kind of fix all the mistakes that we're making. But we usually find a good middle ground. And actually, when you do work together, when you speak to your experimental kind of collaborators, and understand what they want, it can help you write better code. And if the codes kind of explain what their limitations are and what they're looking for, it's actually a lot easier to take better data as well. So it really is a balance that kind of we need to find, but it does lead to really great science when you can do that. Yeah, as I said, happens when we speak the same language. So I mean, this is just an example from our lab. We take images, and as we speak together, we can go, well, actually, as an experimentalist, this is what I'm looking for. This is kind of what I need. I want to be able to find where, for example, a protein is binding on DNA. And then you can speak to the coder and be like, what do I need to get for you to be able to extract this information? Say, well, I need 200 molecules of this and really high resolution. I want loads of data. And you'd be like, well, actually, I can do that. I can maybe get you 100 molecules of this resolution and then kind of come to a nice compromise. And that's kind of all where we always find our best kind of science and analysis happening is when we work really well together. And I think it's very true in this room. Got spanning kind of experimental and computational here. It's really important you all kind of speak together and get a really good understanding of what you actually want from each other in terms of the data set and the analysis.